No, sir.
No, I hope you all are reading your textbooks, the NCERT, and also doing the assignments that we are providing you with constantly, correct?
Sir, I missed the last assignment.
Which one did you miss?
Sir, the last one you submitted.
Okay, so which is the last that you received? What was the topic for that you received?
Sir, I don't remember.
Okay, so just do one thing, reach out to Nishkarsh and tell him that you have received this one and get a confirmation with what all assignments are left, and you will get it, okay?
Yes, sir.
Yeah. Okay, so just a brief, let's just quickly summarize what we have done till now. So because we have started respiration, it's just one class that we have done in it. So we just discussed the basics. We also have Omar joining. Hello, Omar. So we discussed the basics that respiration can be of two types broadly, aerobic and anaerobic. Good evening. Omar is writing in the chat. Okay, so aerobic and anaerobic. Aerobic, which means it needs oxygen. Anaerobic is it does not require oxygen. Now both of them can be of two types, facultative or obligate. If they are obligate something, so if they are obligate aerobic, which means they can only survive in the presence of oxygen, okay? And if they are obligate anaerobic, which means that if there is any oxygen present, then they will die. They can only grow in an oxygen deficient environment. Say the facultative is the opposite of it. Is it clear? Then I told you that we will be focusing on we'll be focusing on aerobic respiration for now, because we have to do glycolysis. And whatever we made as a product, like whatever plants made as a final product of photosynthesis was glucose, which we are now going to break down. Like plants are not going to break down. Glycolysis is happening both in plants and animal cells. So for that matter, every cell breaks down glucose. Now, this aerobic respiration, basically, let me just tell you, there are two parts. Remember, I told you there are certain organisms which are prokaryotic and certain organisms which are eukaryotic, correct? What was prokaryotic and what was eukaryotic? Anyone remember? Just tell me quickly. Anyone? Abhinav, Reba. Prokaryotic and eukaryotic organisms. Mariam? Am I audible? Hello? I hope I'm connected. Can someone confirm if I'm audible to you?
Yes, sir, you're audible, sir. Sir, what was the question?
Oh, okay, thank you. The question was, we have discussed about prokaryotes and eukaryotes, correct?
Yes, sir.
So what is the most important difference between prokaryote and eukaryote, apart from nucleus? So after that.
After that.
Yes, because prokaryote, eukaryote is directly related to nucleus. But if, let's say, nucleus part is removed, what's next? What is the next major difference between a prokaryotic cell and a eukaryotic cell?
I don't remember, sir.
Hey, how can you do this? If you just forgot what we started with at the beginning. We are reaching towards the end of the syllabus, but you cannot keep forgetting what we did. So remember, I told you that in prokaryotic cell, there is no membrane bound organelle present.
Yes, sir. Now I remember, sir.
Yes, no mitochondria, no Golgi bodies. Only ribosomes are common in both because ribosomes do not have a membrane. Remember? They are
Yes, sir.
yes. So in aerobic respiration, basically at the very end, there are two different things that are happening. I told you that in glycolysis, basically we are breaking down glucose to make energy, which is ATP, correct? Correct? Yes or no? Just be with me on the same page. Yes or no? And
Yes, sir.
yes, and ATP, when we are making ATPs, we are generating energy. In a prokaryotic cell, glycolysis happens. In a eukaryotic cell, also glycolysis happens. So glycolysis is common in both the cells because it does not happen inside any organelle. It happens in the cytoplasm, okay, the cytosol. But remember, in eukaryotic cells, we used to call mitochondria the powerhouse of the cell. Mitochondria produces ATPs, right? Yes or no?
Yes, sir. It produces ATP.
But there is no mitochondria in a prokaryote. So how will prokaryotes produce ATP?
Sir, in the cytoplasm.
Yes, through glycolysis. So there is one cycle which is common, which has evolved through time, and that is glycolysis. So we started with glycolysis, which is common in both. So you can write it down. It's common in both eukaryotes and prokaryotes, and it takes place in cytosol. Okay, so this glycolysis happens. Let me just yes. So glycolysis happens both in prokaryotes and eukaryotes, which means that through this pathway, even bacteria can break down glucose to make energy. You do not require any specialized organelle for that. And the reason is because it takes place in the cytoplasm. So both the cells have cytoplasm. Now we started with glycolysis. To begin with, there is glucose, which is a 6-carbon molecule. Just a brief, let's summarize it. The first step in glycolysis is an energy dependent step. And I told you that in the first 3-4 steps in glycolysis, we are actually investing energy, not harvesting energy from it. So they are called energy investment phase or energy required there. So the first step, and whenever in any biochemical reaction, when we commit ATPs, we commit energy, we make that step irreversible. So glucose becomes glucose 6-phosphate irreversibly by using ATP and giving out ADP. The enzyme which helps there is hexokinase in the presence of the cofactor magnesium. Second step is glucose 6-phosphate, which is like there is one phosphate group. And again, so because this is a kinase, it added phosphate. Again, there will be another step where it will isomerize. So glucose will become fructose. From glucose 6-phosphate, it will become fructose 6-phosphate. And now because it's isomerizing, the enzyme will be, the enzyme will be glucoisomerase. And because it's a phosphoglucose, it's phosphoglucoisomerase. Now this fructose 6-phosphate again has to go through addition of another phosphate. So another ATP is used, another phosphate is added, and fructose 6-phosphate becomes fructose 1,6-bisphosphate. And because we are adding phosphate, the name of the enzyme will be a kinase. Okay. And because it's a phosphofructose, it's phosphofructokinase, and with a cofactor called, which is magnesium. Then the fourth step was where fructose 1,6-bisphosphate become glyceraldehyde 3-phosphate. Okay. Now this glyceraldehyde 3-phosphate is a aldehyde. So sugar is converting into an aldehyde, and because of which you see that the enzyme is aldolase and the cofactor is zinc. Now, there is one thing which is this glyceraldehyde 3-phosphate is G3P. Remember? Where have you read G3P before? G3P, people. Glyceraldehyde 3-phosphate. Yes. Who's this? Mariam. Yes, Mariam.
In the Kelvin cycle?
Yes, in the Kelvin cycle. Now, and we also know that there is one more name for it, which is called triose phosphate. Remember? Because there also I use this name, triose phosphate. So glyceraldehyde 3-phosphate is known as triose phosphate. Now this glyceraldehyde 3-phosphate can reversibly in the cell without the use of any cofactor can constantly keep converting and converting back into another triose phosphate, which is known as, so glyceraldehyde. Have you studied these functional groups in your chemistry yet?
Yes, sir.
Yes. So you know that aldehyde, what is an aldehyde? And what is a ketone? So you know aldehyde, ketone, alcohol, carboxylic groups, right?
Yes, sir.
Okay. So this glyceraldehyde 3-phosphate constantly keeps converting into another triose phosphate, which is like the balance between the two is maintained in the cell, depends on the physiology of the cell also. It's called dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Okay, but this cannot, and this is also a 3-carbon thing, and this is also called a triose phosphate. Triose phosphate is it's a bigger group, because any where the phosphate group is present on a 3-carbon chain is called a triose phosphate. So in dihydroxyacetone phosphate also, it's 3-carbon, and it's also called triose phosphate. So both forms of triose phosphate keeps on chemically converting from one to another. But what you have to remember is this acetone part, it does not participate in glycolysis pathway. Is it clear?
Yes, sir.
So you make a star mark here and write down, it cannot undergo further steps of glycolysis. So what do you think, is there any benefit of this step at step 4, if I'm telling you that glyceraldehyde phosphate, which can actually further go into, so I'm making it here, it can further go into the pathway, but if it converts into dihydroxyacetone phosphate, another type of triose phosphate, then it will halt there. So what do you think? Is there any benefit? Is there any benefit for this? Think like a biologist. What if you are under some stressed conditions where your body is very low on glucose, you are starving, you are low on glucose, and remember I told you the first thing that's get broken down for energy in the body is carbohydrates. Next is, suppose we depleted all our carbohydrate reserves. Not all, we'll never deplete it, but suppose carbohydrate reserves are being diminished. What next should we go to? Fats, lipids, perfect, because lipids provide the largest energy from that particular amount per gram. But what is the problem with lipid metabolism? Why don't we metabolize lipids first then?
It's harder to break down.
Yes. And what do you mean by harder? Like the word harder is like it's very hard and we have to hit it harder. Biologically, what's the difficulty in breaking down lipids? Think. When do we burn fat? If I'm sitting, am I burning fat?
No.
No.
But I'm burning, am I burning carbohydrates?
Yes, sir.
Yes. When will I burn fat?
When the
Heart rate should be
carbohydrates
high.
will get over.
I didn't know. Suppose
When
I
you'll
do.
exercise.
Yes. So when I'll start doing something vigorous where I need lots and lots of energy, which carbohydrate cannot provide me. So I shift to another better alternative because now I am giving all that, push to the physiology through my hard work, right?
Yes, sir.
They say that if you are just walking, so sometimes people who are obese, they tend to go on long walks. They are just walking and talking and then they're complaining why I'm not losing weight. Remember I told you very early during our classes that you cannot start breaking down fat if your heartbeat does not crosses over 140 or something, which means if you are not running, if you are not
120, sir.
120 or something, right? Which means
Yes, sir.
if you are not running or if you are not playing. So if you are walking, walking is not very strenuous to your body. It keeps you fit, of course. Walking is a good habit, and if you are walking long, you will get tired, and it's good for your muscles, it's good for your lungs because you'll breathe in open air, but it will not necessarily start breaking down fats, right? To do that, you have to do something vigorous. So next we shift to fats, correct?
Yes, sir.
And that's why, let's suppose you want to halt or you want to regulate glycolysis. So till this pathway, if glyceraldehyde 3 phosphate is there, it will just continue breaking down, like it will continue in the pathway. But if some part of it is getting converted into dihydroxyacetone phosphate, it stays there and that is taken out of the pathway, correct? Stored. It's like you get lots of letters, all of it you have to process. But I tell you that save certain letters, like who are from your very dear ones for later time. Like just keep it away and we'll read it later. Do you understand? So that is one part of regulation that can be imparted from this step. Do you understand? Suppose you are burning lots of fat and at that point of time, you do not want to exhaust glucose. Am I clear?
Yes, sir.
So you keep it as dihydroxyacetone phosphate and do not allow it to go into further degradation. But ultimately, it will come back to glyceraldehyde 3 phosphate and will go further. And from this part onward, I told you that these 4 steps are the preparatory phase or energy investment phase. From this step onward, let's say we go to let's say we go to the next step. Okay. From 5th step onward, we now start getting into a zone where we have to get back the energy that we have utilized. Is it clear? Is it clear?
Yes.
Okay. Now, considering that this dihyd, because this was a 6 carbon molecule, correct? Look here, fructose 1,6 bisphosphate was a 6 carbon molecule. When we broke it down, there were 2 alternatives it can go towards: dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Considering that active glycolysis is happening in your cell, then from now onwards, the 5th step, how many G3Ps will be there? How many glyceraldehyde 3 phosphates will be there?
2.
2, right? Let's consider that it's happening at its full potential. So then from now onwards, there will be 2 molecules per step. Do you understand why there are 2 molecules now per step? Because each molecule is 3 carbon. I have mentioned it here also, correct?
Yes, sir.
Yes. So in the fifth step, what happens is this glyceraldehyde 3-phosphate. Now, if you have a 3-carbon molecule from where you need to take ATPs out, then what it has to lose?
Phosphate.
Exactly. The way you utilize the ATPs, you will get it again the same way. Very good. Brilliant. So this is the step where glyceraldehyde 3-phosphate, which is your G3P, becomes 1,3 first 1,3 bisphosphoglycerate. Do you understand? Now there is one extra phosphate. Here was one phosphate, which was on the 3rd carbon, glyceraldehyde 3-phosphate. Now we have 1,3 bisphosphoglycerate, which is same molecule with 2 phosphates, which means what must have happened? Another phosphate must have another phosphate must have added to this molecule. And why are we doing this? Just understand. There were 2 steps before where we added phosphate, this and this. Now, if we go on to remove the same phosphates, we will get 2 ATPs back, correct?
Yes,
But
sir.
is there any net profit that happened? Any net profit that happened? No, correct. We invested 2 ATPs, we got 2 ATPs out. No benefit, no loss. Why did we this all of that? Or why did we do all this pathway to get no benefit? Now we have to get benefit. But to get benefit, we have to increase phosphate, but we have to increase phosphate without utilizing ATP. Do you understand? Because
Yes.
if we will keep utilizing ATP to add phosphate, then ultimately we will just get the same number of ATPs back when we are removing phosphate. So at this step, I had made this molecule biphosphate. It's a sorry, I just misspelled it. Bisphospho is the word. Bisphosphoglycerate. Okay. So now I have added one more phosphate, but this phosphate is not coming from ATP. So what is the other way it can come from? Which is the another high energy rich intermediate in the cell?
NADH.
NADH, right? Now, NADH is reduced form. Let's understand this. NADH, there is a hydrogen in the end, which means it's already reduced, correct? Correct or not?
Yes.
Yes, sir.
And there is one thing which is actively present in the cell, which is called HPO4 2 negative. You know what is this called? HPO4. What is H3PO4? Phosphoric acid. Is it clear? HPO4 is like biphosphate. HCO3 bicarbonate, remember?
Yes.
HCO3, HPO4. This is biphosphate. Now we have this molecule in the cell and our problem is solved because we have a phosphate. Yes, we can add it. But when we will add phosphate from here, who will take hydrogen?
NAD.
Exactly. NAD is there in the cell. NAD plus. Now this NAD, 2 of these LPs, because there are 2, valency will be 2. So these 2 NADs will take these 2 hydrogens, and when NAD and HPO4 will be utilized, what will it become?
NADH.
Yes, 2 NADH plus hydrogen ion. Okay. But what happened is this phosphate got free and we could add this phosphate because NADH didn't take it. We could add this phosphate to glyceraldehyde 3-phosphate and make it 1,3-bisphosphoglycerate. Is it clear, everyone?
Yes, sir.
Yes, sir.
So, tada, eureka moment. We added one more phosphate without utilizing ATP. And this could only happen because of this molecule. You remember, now do you understand that why this molecule was important and the final product of Calvin cycle as well?
Yes, sir.
Yes. So this molecule here, and to reach to this molecule by breaking glucose, at this first step or the second step, we could not have used NAD. The only way to ensure that we make glyceraldehyde 3-phosphate was to use ATP. So we sacrificed. We said, okay, let me invest. It's like you opening up a company. So someone said that, to open up a company, you need an investment. So first you bring in money, then that money will make more money, because the money that you have used will become something, a service or a product which you can sell and make more money. Correct? Same is happening here. So the way we behave, or society behaves, biology behaves the same. We invested money to make glyceraldehyde 3-phosphate from glucose. Okay. So that finally this glyceraldehyde 3-phosphate can take more phosphates without ATP. Okay. Just from HPO4 in the presence of NAD plus, making NADH and making this molecule as a bisphosphoglycerate. Is it clear, everyone? Now,
Yes.
how many bisphosphoglycerates will be there? 2. Correct? Remember, this is 2.
Yes, sir.
Yes. And now when it is 2, we got an amazing opportunity to do what? So
Remove the phosphate.
yes, exactly. So this 1,3-bisphosphoglycerate can now, oh sorry, let me also tell you about the enzymes. I forgot. So right, from glyceraldehyde 3-phosphate to bisphosphoglycerate, we are adding phosphate. So what should be the name of the enzyme?
Phosphoglycerate kinase.
No.
No.
Kinases are where we are using ATP to add phosphates. What else is happening? Look here. Look here. The process tells the name of the enzyme. What is happening? This
Phosphate
was,
has been removed.
yes. Sorry.
Phosphate has been removed.
Phosphate is not being removed. Phosphate is being added from here, right?
Ah, yes, sir.
But NAD is becoming NADH. But what is being removed?
Hydrogen.
From where?
NADH.
From HPO4. And उसी से तो निकालो.
This hydrogen from the HPO4 is going to NAD so that it can become NADH. So this one is basically this.
Yes, sir.
So what is the other way from where hydrogen is coming? Let me ask you a very simple question in chemistry. Suppose I have a methane. What is methane?
CH4.
Exactly. 1 carbon with 4 hydrogen, right?
Yes, sir.
Now I want to make it methanol. What will I do?
Remove hydrogen, add OH.
Exactly. So to add something, it was glyceraldehyde 3-phosphate, which means on the 3rd carbon, there was a phosphate group added. Now you want to add 2 phosphates on 1 and on 3. So when you are adding 1 phosphate on carbon 1, what are you removing from there? When there is nothing added, what
Hydrogen.
is exactly. So we are basically removing hydrogen from glyceraldehyde 3-phosphate to make it 1,3-bisphosphoglycerate. Do you understand this?
Yes, sir.
So the name of the enzyme will be glyceraldehyde 3-phosphate dehydrogenase. Makes sense to you? Now this is very confusing because people do not expect it to be here. Just like Abhinav said, it's very, very common assumption that, okay, adding phosphate, it's a kinase. So there'll be an option that what converts a triose phosphate or a glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. It's a neat question. And there'll be a kinase because you are thinking phosphate is being added. You will just tick the kinase and feel happy. But remember, there is no ATP involved. Is it clear, people? So no kinases can come in.
Yes, sir.
And always remember that it is a G3P dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase. Write down the full name. I am writing the half name. G3P dehydrogenase is the enzyme. I could have just told you the name, but I thought that let me explain the reason so that you can remember it. Now, is it clear to you why it's dehydrogenase and not kinase here?
Yes, sir.
It's logical, right? It makes sense or no? But because of all this, what we achieved is we achieved another phosphate on that molecule without utilizing another ATP. So what can we do now? So this step, which is the 6th step, is the first step where we get the first ATP out. Now, this 1,3-bisphosphoglycerate becomes 3-phosphoglycerate. Sorry, I forget to change pens over time. 3, now it's not bis, 3-phosphoglycerate. Look, come up. It was glyceraldehyde 3-phosphate. Is it clear? And this is 3-phosphoglycerate. Do you understand, everyone? So it does not go back. So if you add a phosphate in glyceraldehyde 3-phosphate, it becomes 3-phosphoglycerate. Now, which phosphate did we remove? The exact one which we added from HPO4, right? So this phosphate from HPO4 went and got attached to the first carbon, 1, because on the 3rd carbon there was already a phosphate. 3-phosphate, here also it's 3-bis, 1,3-bis. So we remove this one, now it becomes 3-phosphoglycerate. And the best thing is, what do you get in return? Where did that phosphate go? And how many molecules are there? How many molecules are there?
2.
So we can bring in 2 ADPs, which must be present in the cell, and then add this phosphate to that ADP and make 2
ATP.
So just in one step, we got our investment back. How many ATPs we invested during the first 4 phases? 2, correct?
Yes, sir.
So the payoff phase has started and the first installment has already paid all our investment out. So we got ATP here, make a star here. And now what is happening? A phosphate is being added on the ADP. So what enzyme do you think should be here? Again, addition of phosphate, but now ATP is there. ADP or ATP is involved. So now here will be a kinase, correct?
Yes, sir.
Phosphoglycerate kinase.
Exactly. Very good. Phosphoglycerate kinase. How did you thought the name? From here or here? Here, right?
Down, sir.
Yes, because from this molecule, so it will be what do you say?
Glycerate kinase.
Beautiful. So I hope I think you are understanding it now. So no need to mug up. Phospho. You can make sense, right? Oh shit, what's happening? Phosphoglycerate kinase. So now you know when to use kinase and when not to use kinase.
Yes, sir.
Very good. So first payoff phase, we got the first ATP. Hooray. Nice. Very good. Now what should we do to this 3-phosphoglycerate? And remember, they are also 2 in number. Till now, we still have not got any extra energy, which means clearly we yet have to get the benefit, like the profit is yet to come, which means 3-phosphoglycerate will further go into what? we have to make space. Do you understand? So what happens is from this step onwards, again, and remember that we are not committing ATP now, we are making ATP. So all these steps are reversible. Okay. So this 3-phosphoglycerate becomes 2-phosphoglycerate. And what do you think this achieved? Anyone? Any idea? We just removed phosphate from carbon 3 and put it on carbon 4. What happened? Anything happened? Anything happened?
Nothing happened, sir.
Nothing happened. Okay. Okay. And what do we call molecules where just the location of the functional group is different, or just another molecule is different, but it's the same molecule otherwise? What do we call them?
Positional
What?
isomer.
Positional what?
Positional isomer.
Isomers. Not all the time. Here, okay. So there is something called an enzyme called mutase. Mutase basically just changes the molecule, okay, slightly, and certain properties of it. Have you also studied in chemistry the properties of molecules, chirality and things like that? Chiral molecules, achiral molecules,
No, sir.
light rotating molecules, nothing of that sort you have studied till now. Okay, so let me not go into the detail, but just remember there the enzyme which is there is a mutase. And since it is a phosphoglycerate which is mutating, so the name will be
Phosphoglycerate mutase.
yes, phosphoglycerate mutase. So it's just kind of mutating or changing the molecule. In biology, the word mutation we use, na? What is mutation? A change in the gene, right?
Yes, sir.
Yes, sir.
So you can just remember it through that. The mutase changes, phosphoglycerate mutase changes 3-phosphoglycerate to 2-phosphoglycerate. It's still phosphoglycerate. And they are also, they are also 2 in number. Yes, Abhinav, you wanted to say something?
No, sir.
Okay. Now, I asked you, what do you think it achieved? Do you think it achieved anything? Sometimes the presence of a phosphate group or a functional group on the terminal carbons, because it's a 3-carbon molecule, phosphoglycerate. Now, on carbon 1 and carbon 3 are the terminal carbons, correct?
Yes, sir.
Chemistry. So to change that molecule into something else or to add something else, we might want to bring that bigger chunk on the center molecule so that the free end, the terminal carbons are free. And if you know by now, just if you don't know, just remember that when the terminal carbons are free, they are the most potential sites for reactions. Because what is the benefit with terminal carbons? Have you all studied about steric hindrance in chemistry yet? Steric hindrance. Have you studied steric hindrance?
Yes, sir.
What is steric hindrance? The word steric hindrance. What is it?
It moves.
It is if you have too much crowded molecules around a carbon, too much big molecules, phosphate, because phosphate will not be just P, na? Phosphate itself is a big molecule. It will be having other hydrogens and oxygen with it. So it will not allow any other reactant to reach the carbon core. That is called steric hindrance. It's just like you all are like standing, like covering me and very protecting. Yes. So you are just preventing. It's more than protection. It's hindrance. So the reacting group is now not reaching the carbon core to react with it. Is it clear?
Yes, sir.
So the benefit with making it 2-phosphoglycerate is now, because from 2-phosphoglycerate, we can easily make it phosphoenol pyruvate. phosphoenol pyruvate. Remember that all of them are 3 carbon molecules. That's why I'm writing into 2. But from 3 phosphoglycerate, it's not possible to make phosphoenol pyruvate. So you convert it into 2 phosphoglycerate first. So you can make it phosphoenol pyruvate. And this is a very special molecule. I'll tell you why. But let's first guess the name of enzyme. Now, what is happening is you are adding an enol. You are making it an enol form. From phosphoglycerate, we are making it phosphoenol pyruvate. So what will be the enzyme? When we were making something from a sugar to aldehyde, we were using aldolase.
Ase.
Here we will
Enolase.
Enolase.
Exactly. Perfect. You people, have you studied it already or you're just guessing it?
Guessing.
Guessing, sir.
You are brilliant. It's called enolase because we are aldolase, enolase. And this particular enzyme uses either magnesium. Some books say that it uses magnesium, but let me tell you, it also uses manganese. Okay, Mn 2+. So both are correct. So if this question comes that what is a cofactor for enolase, if Mg and Mn both are in the options, there will surely be an option both of the above or none of the above. So you have to choose both of the above. Is it clear?
Yes, sir.
Yes. Mostly people think that enzymes can use just one. So of course it is using one at a time. It's not using both. So it's always magnesium or manganese. It's not magnesium and manganese. Okay, but any of the two can be used, but only one at a time. Is it clear, everyone?
Yes, sir.
So enolase converts phosphoglycerate into phosphoenol pyruvate. And when this was happening, actually we just removed water out of it. So to convert a phosphoglycerate into a phosphoenol pyruvate, you have to remove H2O. So you can just write, because there are 2 molecules, so there will be 2 H2O. Just write it, this we remove. Okay.
Yes, sir.
Okay, sorry. So now we have phosphoenol pyruvate. And I told you that phosphoenol pyruvate is a interesting molecule because this phospho group, which is present in the pyruvate, which is present with the pyruvate group, can be lost. And this gives us a benefit. We have just one phospho left, right? Per molecule.
Yes, sir.
Per molecule, we have just one phospho left. Now, we think that, and this phospho was present here also. So if you actually can feel chemistry, like I always tell my students, that whenever you are seeing in a biochemical pathway, that things are just very boringly changing from one thing to another, it means that molecule is itching to do something. Have you seen a restless person who wants to go out, but you are not allowing that person to go out? So what that person will do? It will just change positions in the same place, right? A very calm, composed person will sit in the chair and will be like, okay, I don't need to move. Stable. But if that person wants to do something, you're not allowing that person to do something, that person will just keep changing positions, right? It will look right, left, do something, and we call that this person is very restless. It's not sitting peacefully. Correct or not?
Yes, sir.
Same is with biochemistry. These 3 phosphoglycerates, first what the molecule did nothing. That did not make ATP, ADP did not utilize anything, just change the position. Then again changed the molecule, again did nothing, just remove some water. These 3 steps clearly tell us that this is being restless and it really wants to get rid of this phospho, because once it get rid of the phospho, it can make ATPs, correct?
Yes, sir.
And this ATP will be a profit ATP because compensation has already happened. So first it changed the position, could not get rid of phospho. Then it changed the molecule, which shows that now it can get rid of the phospho because pyruvate can be alone. So here again, because there are 2 molecules, so you use 2 ADP in the very final step. and you make
2 ATP.
ATPs. In the last step, we get that profit that we wanted, for which we were itching. The pathway was itching from last 3 steps. कि अभी मैं क्या करूं? Like, let's change 3 phosphoglycerate to 2 phosphoglycerate. Okay, कुछ नहीं हुआ. Let's change 2 phosphoglycerate to phosphoenol pyruvate. It's just preparing itself to make to convert into a molecule which can easily let go of phospho, so that we can make 2 ATPs, which is a profit ATP. And now what will happen? What will be the result? This phosphopyruvate becomes pyruvate. It can stay alone. Now you tell me, and again it's a 3 carbon molecule, so it will be 2 in number. Now you tell me what was happening here. Phosphoenol pyruvate became pyruvate and ATP was mode. So what should be the enzyme's name?
Pyruvate kinase.
Pyruvate kinase.
Exactly. See, people are perfect. Pyruvate kinase. Beautiful. And because it's a kinase, it will use magnesium 2+. Also, pyruvate kinase is a very, very important enzyme, because pyruvate is an important enzyme. So sometimes it also uses, so I'm just writing or, it also uses potassium. Okay, K+ as a cofactor. But mostly the major one is magnesium 2+. It has been the major one through the pathway at multiple times. So if a question comes, which micronutrient is the most important cofactor from the perspective of glycolytic pathway, then the answer is
Magnesium.
Magnesium, because it's also working for pyruvate kinase, for enolase. It's also working for your phosphofructokinase, and it's also working for hexokinase. Clear?
Yes, sir.
Yes, beautiful. That's why it was important. So now we have pyruvate, and this phosphorylation process, this last step, this one here, where we made the ATPs. So you can write the profit ATPs here. Per glycolytic cycle, when you break down one glucose into 2 molecules of pyruvates, what is the net ATPs that are produced? Tell me. What will you choose as an answer? 2, 4, 6, 8 are the options.
Six.
ATP produced. Just count the ATPs. Where are 6 ATPs produced?
Is it 2 molecule of ATP like that?
Yes. So in one glycolysis cycle, when you break down one glucose to make 2 molecules of pyruvate, what is the net production or net synthesis or net profit of ATPs in number? How many ATPs do we get out of glycolysis pathway?
Can you repeat, sir?
Okay. How many ATPs do we get out from the glycolytic pathway?
10.
Let's
Sir,
start.
NADP will also convert, no?
What?
Sir, NADP will also get into ATP, no?
Where is NADP used?
2 ATP.
There is no NADP used in glycolysis at all. Let's go from step 1. Now look here, Abhinav. This was a glucose, right?
Yes, sir.
You use the ATP, one single ATP, to convert it into glucose 6-phosphate. So you can do one thing. Keep both your hands like this. Your left hand will count for investments and your right hand will count for profits. Is it clear?
Yes, sir.
So let's start. At first step, you have one ATP invested to make glucose into glucose 6-phosphate, correct? And please,
Yes,
everyone pay attention, don't get confused. Then you changed that glucose into fructose through isomerase. That again at the third step used another ATP to make fructose 1,6-bisphosphate. So 2 ATPs are invested by now. Net profit is still 0, right? Nothing got out. Then in this 5th step, you actually use the NAD to take this hydrogen from HPO4 biphosphate and use this HPO, utilize this phosphate group, PO4 group, and attach it on this glyceraldehyde 3-phosphate and make it 1,3-bisphosphoglycerate. Is it clear, everyone?
Yes, sir.
NADPH is yet not used. There is no concept of NADP or NADPH here. Because
Yes, sir. I understood, sir. I got confused here.
yes, because if NAD will take P and become NADP, then this whole pathway will just fail. It's like NAD ditched you, backstabbed you. You wanted NAD plus to take hydrogen and get reduced and get lost and leave the phosphate for glyceraldehyde 3-phosphate. Instead, if your NAD will take P and become NADP and go, then you're just robbed, right?
Yes, sir.
Yes. So NAD plus comes in with HPO4, takes the hydrogen, gets reduced to NADH and goes. This PO4 gets added to glyceraldehyde 3-phosphate and it becomes 1,3-bisphosphoglycerate. And these two hydrogen has come out from this position where PO4 got added. So it's an enzyme is called G3P dehydrogenase. Then the first step is where you actually produce 2 molecules of ATP by removing the same phosphate which got added in the previous step. From 1,3-bisphosphoglycerate, you just make 3-phosphoglycerate and removed 1 phosphate from each molecule and put it in 1 molecule of ADP each. So you make 2 ATPs. So you had 2 investments till now. You got 2 profits, but which got compensated. So the net is still 0, correct?
Yes, sir.
Then you were itching to make profit. You are like, okay, my investment is recovered, but I am an entrepreneur. I have to get profit. Otherwise, I'm a failed entrepreneur. And if I fail, that cycle will fail. The cell will die. So you are itching. So you are changing position of the phosphate. You are changing the molecule. Finally, you made phosphoenol pyruvate, which could lose phospho and again add it to the 2 ADPs and make 2 ATP. Now these 2 ATPs are the first profit you had in the whole cycle, and the cycle ended at pyruvate. Is it clear?
Yes, sir.
So let me ask you again, what is the net profit of ATPs in one cycle of glycolysis?
4?
2.
Sir,
2.
2.
Yes, sir. 2.
I will hang you upside down, Abhinav, with your toes.
Profit, sir. I got confused there also, sir. Profit.
Yes.
2, sir.
Yes. So they always ask, remember, if you read the question properly, you will not commit mistakes. If the question comes, how many total ATPs are produced? Total ATPs are produced in glycolytic pathway. Then it is 4. Because at 2 different steps, 2 plus 2 ATPs are produced. How many are used? 2.
2.
In step 1 and step 3, which is the preparatory phase. So preparatory phase takes 2 ATPs and payoff phase gives 4 ATPs. So what is the net? If the word net is there, what is the net profit out of one cycle?
2 ATP.
2 ATPs. Is it clear?
Yes, sir.
If they ask you one glucose molecule which undergoes glycolysis gives the cell how many ATPs?
2.
Overall, 2. Is it clear? Because
Yes,
what
sir.
came out?
Sir, if they ask overall, then
No, but from the whole pathway, na, what came out that cell can utilize for the cell? You have to read the question carefully. What came out for the cell to utilize?
yes, sir.
2, right? And if they ask you at how many steps are there in which ATPs are produced, then there are 2 steps where ATPs are produced. One where you remove this, where you convert 1,3-bisphosphoglycerate to 3-phosphoglycerate, and the other is when you convert phosphoenol pyruvate to pyruvate. At these 2 steps, ATPs are produced. And at which 2 steps ATPs are used? You have to remember those 2 steps also. The first step, glucose to glucose 6-phosphate, and then fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-bisphosphate. Will you remember these certain things now?
Yes,
Yes,
sir.
sir.
So finally we get pyruvate, and the enzyme is pyruvate kinase. And this last step where we actually made this ATP is called, both the steps actually where we produced ATPs are called substrate level phosphorylation. There are different types of phosphorylation. Remember everyone? One phosphorylation we studied in Calvin cycle, which is called photophosphorylation, right? Right or no?
Yes, sir.
Because it was happening because of photic energy, the light energy. This is called substrate level phosphorylation because molecules are interacting among themselves and from one substrate it goes to another substrate. So this step, the last step, and this one also, the both are called substrate level phosphorylation. This is something very high end, but it has been asked. So that's why I'm telling you that in glycolysis, ATPs are produced through substrate level phosphorylation. Whereas in in the Z scheme, remember the Z scheme? Remember the C3 cycle, the Calvin cycle? Electron transport chain, do you remember that or not? Just above.
Yes,
Yes,
sir.
sir.
Remember, I told you that in a Z scheme, there is also a ATP produced, right?
Yes, sir.
But that is not through substrate level phosphorylation. That is through photophosphorylation. Is it clear?
Yes, sir. Clear.
Phosphorylation. Okay, just write it down. So this is the whole pathway of glycolysis where we put in glucose and we get out 2 molecules of pyruvate. Any doubt in glycolysis, everyone? It's a very, very important pathway. Simple but important. Once you understand it, you will score marks. Once you get confused, you will lose a lot of marks. Preparatory phase, payoff phase. Total, there are 10 steps or like 5. So this is 6th, 7th, 8th, 9 steps basically. So total there are 9 steps. Just go through all these steps. This step is just a conversion, like here. Oh, sorry. So glyceraldehyde 3-phosphate, when it is converting to dihydroxyacetone phosphate, what is happening? An aldehyde is converting into acetone. Acetone is a ketone, right? Aldehyde to ketone is called isomerization. Remember in chemistry?
Yes, sir.
So what should be the enzyme? There is also an enzyme here. So let's keep it out. So let me make it like this. I'm taking this cursor here and we are writing the enzyme name here. So there is an enzyme here. Can you guess the name? Remember, a triose phosphate, which is a glyceraldehyde, is being converted into another triose phosphate, which is a acetone. So it's isomerizing. So what should be the name of the enzyme?
Aldolase.
This is aldolase. From a sugar, fructose 1,6-bisphosphate.
Triophosphate isomerase.
Exactly. It's called isomerase, and because it's happening with a triose phosphate, both of them are triose phosphate. Okay. So it's called a triose phosphate isomerase. And since it is a reversible pathway, this enzyme can do both. It can convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate and also do the reverse. Is it clear, everyone?
Yes.
Yes, sir.
So write down triose phosphate isomerase. Isomerase. It is isomerase, right? Triose phosphate isomerase. Yeah. Okay, I'm giving you one homework. I know it has not been asked. It's for me. Will you do one thing for me?
Yes, sir.
You have to find what is the cofactor for this enzyme. Will you do this?
Yes, sir.
Yeah. Okay, so this is the complete glycolysis pathway. Before I ask you the question, the next question I'm going to ask you is, what happens to this pyruvate now? Is it just done? Just throw it out or something else? Because
Aerobic, anaerobic.
yes, very good. So I give you 3 minutes, 2 minutes. Just look at this whole glycolysis pathway and let me know if you are thorough with it so that I can move forward. Take your time and allow me, I'll fetch myself a glass of water. Okay. Is it fine? 2 minutes?
Yes, yes.
Yes, sir.
Go through the pathway. Let me know if there is anything which is problematic or you don't understand. I'm just coming back, okay? Hello, everyone. Everyone is back.
Yes, sir.
Okay. Any doubts? Any problem in the glycolysis pathway?
No, sir.
No. So let's move forward. Now, if I ask you before we went on the break, Mariam just used certain words. She said aerobic and anaerobic pathways, right? Mariam, you said that.
Yes, sir.
So let me ask you, what do you think glucose is a part of aerobic or anaerobic? Which pathway? Glycolysis.
Aerobic.
Sorry?
Aerobic.
Aerobic. So do you see anywhere in the pathway oxygen being utilized or used or coming in?
No.
No, right? Which means glucose happens in all the cells, whether they are aerobic or anaerobic. The starting of respiration, the starting of formation of energy starts from glycolysis. You take glucose, you start breaking it down through glycolysis in the cytosol, and you overall make 2 ATPs as a profit. Is it correct?
Yes, sir.
Which means that any given cell on the planet Earth, bacteria, prokaryotic, eukaryotic, fungi, human, plant, at least how much ATPs from one glucose it can secure? 2, right? 2 is very less. 2 ATPs per glucose is very less, correct? Correct or not? But this is the minimum that every cell can ensure, even if it is doing an oxygen independent respiration, because no oxygen is utilized in glycolysis. Is it correct, everyone? Is it correct or not?
Yes, sir.
Yes. Now write down let's write down certain things about glycolysis that has been asked in entrances or boards. The energy yield from one molecule of glucose, the minimum energy yield, write down the minimum energy output or energy yield, whatever you want to write. The minimum energy output from one molecule of glucose in glycolysis is 2 ATPs, right? 2 ATPs and 2 NADH. Abhinav, I want you to pitch in. You used prematurely something about, you were saying something about NADH, right?
Yes, sir.
But that thing is relevant now. So is NADH an energy molecule?
Yes, sir.
Can you use NADH in your body?
No, sir. It gets converted to ATP.
Yes, but till now, is it a energy currency?
No, sir.
It's a check or a demand draft. Now for both check and demand draft, you have to depend on a factor which is called bank. Correct?
Yes, sir.
To produce ATPs from NADH, you have to depend on oxygen. Is it clear, everyone?
Yes,
now,
sir.
This is where the oxidative part or the aerobic part comes in. Glycolysis is basic. It happens in every cell, whether it uses oxygen or it does not use oxygen, does not matter. At least with every glucose, 2 molecules of ATP will be produced along with 2 NADH. So write down the sentence, complete the sentence. The output is 2 ATP molecules and 2 NADH molecules. Now this NADH molecule is not of any use to the energy currency if there is no oxygen. It will be just there. So a prokaryotic bacteria can only use that ATP. Okay, is it clear?
Yes, sir.
A prokaryotic anaerobic bacteria can only use that 2 ATP. And I think 2 ATPs per glucose molecule is sufficient for a bacteria, correct? To just survive?
Yes, sir.
Single cell, right? Yeah, it's fine. But suppose you have oxygen, you are aerobic organism, then you have some additional benefits from this glycolysis pathway. You got 2 NADH also. And these NADH can now undergo what we say as oxidative phosphorylation. Okay, so write down the NADH, which is 2 NADH, which comes from glycolysis, can undergo oxidative phosphorylation. Photophosphorylation, substrate level phosphorylation. The third one is,
Oxidative phosphorylation.
what does the word oxidative phosphorylation tells you? That it is dependent on
Oxygen.
photo was dependent on light. Substrate level phosphorylation was dependent on two molecules colliding together and doing the chemical reaction. Oxidative phosphorylation is dependent on
Oxygen.
So 2-NADH can undergo oxidative phosphorylation.
लग रहा है सबको पेट खराब चल रहा है।
Aisha. Is it Aisha? Oh, is it Aisha? Aisha is she somewhere else completely? Are you there, Aisha?
I'm sorry.
It's fine.
Actually, my brother, he was just disturbing me.
Make sure you're there. Okay, so we have 2-NADH, which upon oxidative phosphorylation can give us a total of around, it's very weird, but around
3 ATP.
no, not exactly 3 ATP. Do they use 3 ATP in your NCERT?
Yes, sir. They have given, like in the equation, 2 into 3 is equal to 6 ATP. So 1 for 3 ATP.
2 into 3 is equal to 6. So from oxidative phosphorylation is getting 6 ATPs. But in reality, actually, two molecules oxidize together. And from 2-NADH, we actually get 5 ATPs. So 2.5 per molecule, which does not make sense. There cannot be a half ATP, right?
Yes, sir.
But the crux here is that 2-NADH together for they get oxidized for phosphorylation. It can never happen as one. So we get 5 ATPs. Now, if NCERT says 6, you should remember 6 for your entrances. But know that in reality, it's like 5, somewhere around 5. Okay. Because 2.5 per, I think they have rounded it off. So let's also round it off to 6 ATPs. Okay.
Yes, sir.
Because NEET follows NCERT if you're giving NEET. But you remember that in reality, what we have discovered now, it's less than 6 ATPs. It's actually 5 ATPs per 2 molecules, which means 2.5 each, which does not make any sense. Is it clear? But it happens in couple.
Yes,
Now, if I ask you, let me rephrase some questions from entrance exams. In a prokaryotic anaerobic bacteria, the result of glycolysis, as a result of glycolysis, how many ATPs do the cell get?
Get
2. Whenever I say the cell get, means the cell is getting it to use.
2, sir.
2. If I tell you that in the presence of oxygen, what is the total number of ATPs that come out of glycolytic pathway?
10.
8. According to
Come out. Yes, sir.
when I'm saying come out, of course, coming out of that pathway is for the cell to use.
Yes, sir.
So this 6, which is coming from 2-NADH
Plus 2 ATP.
plus 2 from the actual pathway. Everyone is following or not? See, things are getting confusing, but if you are with me, I think I'll explain it to you the way you will understand it better. So 6 plus 2, the total output, or in exams, it has also been asked, what is the maximum possible output of ATPs from one cycle of glycolysis? The answer is
8.
ATPs. So you can maximally get 8 ATPs from glycolysis under aerobic respiration. Under? Yes.
Sir, in anaerobic, like in the anaerobic bacteria, you told, then what happens to the ADH there? Because they'll also produce it, no? Because they're going through the same cycle.
What happens to the NADH?
Yes, sir. NADH there.
Nothing happens to the NADH. They cannot do it. They cannot utilize it to make ATP.
So they remove it from their body, or they just keep it there.
Probably, like, I have to go back and read myself that do they have some other pathways there they put this NADH for some other use, because of course it's a good molecule. It's having potential in it. It's high energy rich intermediate. So if you cannot convert it into ATP, I don't think that you will just throw it off. It's like something came to your house. You cannot use it for something, but you can reuse for something else, right? I'm sure that it must be doing something to it, if not producing ATPs out of it. But I have to read. So I'll get back to you about this question. Is it okay, Abhinav?
Okay, sir.
Yeah. But yes, NADH will be there, but it will be of no. But so therefore, anaerobic prokaryotic bacteria can only make 2 ATPs per glycolysis cycle. So anaerobic bacteria has to go through other ways, other pathways. It cannot only rely on glycolysis. Do you understand that?
Yes, sir.
Yes. So we'll talk about that later, anaerobic. But do you understand about glycolysis completely? In the presence of oxygen under aerobic respiration, the maximum it can produce is
8
ATPs. So this will be your answer. Cool. And by now you have studied about, and if there is also a question, I have just written it here. Yeah. So the question was, in a aerobic respiration through glycolysis, how many ATPs are produced through oxidative phosphorylation and substrate level phosphorylation respectively? Now the option will be respectively. So how many are coming from oxidative phosphorylation?
6.
6. And how many from substrate level phosphorylation? 2. So this is just another way to make that question hard. The question is asking basically the same thing, but it's breaking it down, that how many are coming from oxidative and how many are coming from substrate level. So the answer will be 6 and 2 respectively. Okay.
Yes, sir.
According to NCERT. In reality, it's 5 and 2 respectively, but fine. So that's all you needed to know about glycolysis. Anything else? So you can just write one more line to clarify things. Just write down glycolysis in the absence of oxygen. Glycolysis. So this was in the presence of oxygen. Glycolysis in the absence of oxygen produces only 2 ATPs. Produces only 2 ATPs through substrate level phosphorylation. Is it clear, everyone?
Yes, sir.
Substrate level phosphorylation. Now, let's talk about the byproduct of glycolysis, which is pyruvate. Okay. So what happens to this now is the question. Should we just, because now it's fine, so let's throw out pyruvate. No, pyruvate is another energy-rich intermediate. It still has a potential to go into making more ATPs for us. But how? How should we do this? So clearly, as pyruvate, it cannot do it. So here comes the part which is called the mitochondrial respiration, or the aerobic respiration happening in mitochondria. The reason because of which we call mitochondria the powerhouse of the cell. We do not call cytosol the powerhouse of the cell. Now, one thing is very clear. If we are calling mitochondria the powerhouse and not the cytosol, because your cytosol, like Abhinav cytosol, in an aerobic glycolysis, how many ATPs are producing? 8. Right? Right, Abhinav? Right or not?
Yes, sir.
And cytosol is a very big field. It's like the whole thing in the cell is cytosol. But if we are still calling mitochondria the powerhouse, it clearly means that mitochondria through some pathway is producing per pathway more than 8 ATPs. Is it clear? Make sense?
Yes.
That's why we rely on mitochondria, right? That's why sperms, remember I was teaching you about sperm, I told you that sperm has a head. Something not relevant, but I'm just diverging a little. So in the head part of the sperm, there is something called acrosome. So this top part, you are fine with these things where I digress a little and tell you things, right?
Yes, sir.
Okay. Even if you are not fine, let me tell you that you have to do it. I am teaching. It's for you. And we have a tail. So this is called acrosome. Just remember it. Acrosome. In 12th, you will be reading in detail about sperm. So just know it now. And then there is the head part. In the head part is the DNA, genome. And this genome will be haploid or diploid? Haploid, diploid, forgot everything. Come on. This is a sperm, it's a gamete. So it will be having single copy, right? Because the other one is coming from the mother, ovum. Correct?
Yes.
So in the head part is your genome. Then you have a neck part and a tail part. So tail is clearly the microfilaments, intermediate filaments, basically, which are like flagella, which are helping it to move like a propeller. This neck part has a series of mitochondria arranged in a series. It's like a batteries stacked one below the other to give all that power required for that tail to whip and whip vigorously so that the sperm can move at a cruising speed. Do you understand?
Yes, sir.
So all the neck part contains is lots and lots of mitochondria. Lots means there's a fixed number per sperm, but it contains mitochondria. I just told you because it's important there.
Sir,
Yes, Abhinav.
so the number of mitochondria in all the sperms will be same only?
Yes, in all these sperm necks, the mitochondrial number is same. And if I'm not wrong, that mitochondrial number is, I have to figure out. Like, this is a factual question that they sometimes ask. I don't see there is a relevance, but you can go and figure out. Will you figure out for me, Abhinav?
Yes, sir.
How many mitochondria are present in each sperm's neck? And also figure out if it is fixed all the time or are there any exceptions. First, find for human sperm, because we know for human sperm, they ask for human sperm. I think in different species sperm, it will be different. But find for human sperm, is it same all the time or is there any, what do we call it, exception? Will you do it? Number?
Yes, sir.
Cool. So tomorrow we'll discuss about it. Now this pyruvate, which is here, it goes into the mitochondria. Where does it go?
In the mitochondria.
And in mitochondria, what happens to it is further oxidation. The process is called pyruvate oxidation. So write down the pyruvate in the presence of oxygen. The pyruvate. So I'm not following exactly NCERT, I believe, because NCERT after glycolysis goes to fermentation, I think. If someone can.
Yes, sir.
And then it goes to the this Krebs cycle. But I think if we have started one thing, let's just finish that molecule. Let's trace that molecule to the end. Then we'll come back to fermentation. Is it clear? Is it fine for you?
Yes, sir.
Yes, fine. So we already had pyruvate. Let's say this pyruvate goes into mitochondria and gets further oxidized. So write down, in the presence of oxygen, pyruvate oxidation occurs. Pyruvate oxidation occurs in the mitochondrial matrix. In the mitochondrial matrix. Matrix means?
Liquid.
Yeah, inside. Like there is a cytosol inside the cell, cytoplasm. There's a nucleoplasm inside the nucleus. Then there is a mitochondrial matrix inside the mitochondria.
Like the mitochondria.
Okay, inside. So, which means if let's make a mitochondria, and you all know it's a double membrane.
Membranous structure.
But the second membrane is not straight. Okay, this is a very good question that came to my mind. It has been asked. Which membrane is bigger, the inner mitochondrial membrane or the outer?
The inner, because it is coiled up.
Exactly. It's very counterintuitive to think that the inner mitochondrial membrane is many a times larger than the outer mitochondrial membrane. And whenever you have to fit a larger thing in a smaller thing, the only thing you can do is to fold it, right?
Yes, sir.
Yes. So the inner mitochondrial membrane is not coiled, use the word coiled. I will say that it is folded. Multiple folds are there. So it's like this, right? You always make mitochondria like this. So if you straighten this up according to mathematics, which line is larger?
The one which is inside.
Yes, this is a longer line. So the inner mitochondrial membrane is actually bigger. And inside this, pyruvate undergoes, this is called matrix. Okay. The space between outer membrane and inner membrane is not called matrix. The space inside the inner membrane is called matrix. It is, what is this called then? Intermembranous space between two membranes. Okay. Interschool
Yes.
competition, right?
Yes, sir.
Yeah. So this is matrix where pyruvate undergoes pyruvate oxidation and becomes what? Oh, I should. So let me. Okay. So this happens in mitochondrial matrix. Let me write it here. And pyruvate becomes. Any idea what it becomes?
Acid?
What?
Pyruvic acid?
We are oxidizing it. So it becomes a molecule. Remember the name from now onwards. You know, get a tattoo of this name if you can. It's called acetyl coenzyme.
Yes, sir, I remember this.
Acetyl
It's there in further chapters also.
Yes. Further means 12th, right?
Yes, sir.
Yes, very good, very good. Acetyl CoA. Pyruvate becomes acetyl CoA. And from here onwards, whenever I say acetyl CoA, remember that what is the lineage? It came from glycolysis. It was a pyruvate which upon oxidation became acetyl CoA. And it is because it's important. So let me also tell you the structure. It's very simple. See, what is pyruvate? It's a 3-carbon molecule. So let's make 3 carbon. And of course, this will be hydrogen. And then on pyruvate, both the groups have, sorry. Pyruvate is a very good molecule. 1, 2, 3. Okay. CH3. No, sorry. This should be it. I just made it by mistake. Yes. It's not possible na here, right? According to chemistry. And then you have a oxygen atom at the end. Okay. So this is your pyruvate. This whole molecule is your pyruvate. No, this is not important. It will not be asked, but I'm just telling you how pyruvate converts into acetyl CoA so that you can realize and remember that it's actually a pyruvate only. But what happens? There is something called coenzyme A, CoA, inside the cell. So this pyruvate reacts with coenzyme A, CoA, and in the presence of what basically it's called as pyruvate dehydrogenase. Yes. Pyruvate. Just wait 1 minute. My laptop is on low battery. Yes, fine. Pyruvate dehydrogenase. It's important in 12th, but not right now, but just know it. Pyruvate dehydrogenase, and it's a complex. It's like a complex of enzymes. And we still, if it's a dehydrogenase, it's clearly telling that what it will do to this complex, it will remove
Hydrogen.
hydrogen out of it. So when it will remove hydrogen, someone has to accept hydrogen. Yes or no?
Yes, sir.
And what will accept hydrogen? What accepted hydrogen in glycolysis? A good molecule that can accept hydrogen.
NAD.
Exactly, and still be useful, right?
Yes, sir.
So A will be capital. So NAD plus is there, ready to accept hydrogen. It will come, accept hydrogen and become.
NADH.
So just by oxidizing pyruvate, you did two things. One, you made pyruvate into a acetyl CoA. So the acetyl CoA is simply CH3 carbon with oxygen. And now you have a sulfur with a CoA. Okay. And as a result, what you lost are some hydrogens to NAD, and also you lost a carbon which becomes CO2. Is it clear, everyone?
Yes, sir.
So this molecule here, CH3C double bond O SCOA, this coenzyme. So this COA coenzyme A is itself a molecule, but if I draw that also, it will become more hodgepodge and complex. So just write it at COA, coenzyme A. Is it clear?
Yes, sir.
I'm not going into that detail, but this together, now there are how many carbons?
2.
2 only. So from a 3 carbon pyruvate, we converted into a 2 carbon, and 2 carbon, then that's why we will use the name acetone. Remember?
Yes, sir.
Yes. Acetic acid.
Yes, sir.
So it becomes acetyl because it's tyl
Choline.
CoA, acetyl CoA, acetyl coenzyme A. Plus CO2 is released because we lost 1 carbon. Okay, cool.
Yes, sir.
Now this process helped us with 2 things. First, it gave us acetyl CoA, and this acetyl CoA is an important molecule because it can go into Krebs cycle. Just like Calvin cycle in plants, in respiration, we have a Krebs cycle. Okay.
Yes, sir.
In aerobic respiration. So this acetyl CoA will go in Krebs cycle, but we also got NADH. And what is the benefit of getting NADH?
ATP.
We can further do oxidative phosphorylation of NADH and can convert it into
ATP.
ATP. Very good. So dual benefit, right? So make star in front of this and make a star in front of this. And this is what we achieved from pyruvate. So pyruvate goes into mitochondria and becomes acetyl CoA, releases CO2 and also gives us NADH. Is it clear?
Yes, sir. Clear, sir.
Okay, now write down certain things. This pyruvate, this acetyl coenzyme A enters the citric acid cycle. So there are 3 names which essentially mean the same thing. Okay, either you call it, so I'm making a line here because we are good. Okay, I'm using another color. So either you call it citric acid cycle, which clearly tells us that citric acid will be a part of it, right? And where is
Yes, sir.
citric acid present? Common source for citric acid is natural sources.
Oranges.
Citrus.
It is
All
fruits.
fruits, yes. Lime, lemon, oranges, moshambis, all these things. So citric acid cycle, or you call it tricarboxylic acid cycle. Tricarboxylic acid cycle, or you call it Krebs cycle. Okay. Krebs.
Yes.
They all are the same thing. Just like you call it Calvin cycle or C3 pathway, it's the same thing, correct?
Yes, sir.
So Krebs cycle. So Krebs was the person who discovered it. So write down This cycle was discovered by Krebs. Krebs, he was a German biochemist. Okay. And he also got Nobel for this. Because this is a very important pathway which tells us about all our energy productions in mitochondria. Why mitochondria is powerhouse, this cycle explains. So Krebs, German biochemistry, he got a Nobel in 1950s. I think it's 53 or 4. Will someone confirm for me? But he got a Nobel in one of the year. That year is important. Sometimes I do not remember the years, but just confirm this, okay? I know that it's in the mid 50s, but which year it is, I'm not sure. But he got a Nobel for it.
1953, sir.
Sure?
Yes, sir. On Google.
Okay. Just re-verify it, but most of the time it will be fine. So what is it? 90?
53.
53. You used Google. Wow. You're keeping a Google aside, kya? Is this the reason you were giving me all the answers for the enzymes?
No, sir. Only this one I Googled.
Okay, fine. 1953. Okay, so Nobel Prize he got for it. Okay. Now write down certain things about this cycle. And this is a more complex cycle. We'll start and finish it the next class. But write down. This cycle takes place. This cycle takes place in the matrix. In the matrix of mitochondria. This cycle takes place in the matrix of mitochondria. Okay. This cycle takes place in the matrix of mitochondria. And another direct question is, okay, that I will say in the end. Let me finish the cycle. So first write down, there are 8 major steps in Krebs cycle. There are 8 major steps in Krebs cycle. And whenever I say any pathway cycle, what does it mean, Abhinav? Was glycolysis a cycle?
No, sir.
It was a pathway, a linear pathway. But I'm telling Krebs cycle is a cycle. So what does it mean?
The end product can be converted into the first product?
Yes, the end product will be converted back to the product which will react with acetyl CoA. So the whole cycle will just keep on going. That's why it's a cycle. So write down, there are 8 major steps in the pathway. And I'll just dictate you the steps today. If I start with the steps today, I think we will not be able to finish it today. So let's take this whole pathway tomorrow and finish it. Okay.
Yes.
This is a very important major pathway. It's also very long in terms of explanations. I have to explain what happened. The biochemistry of glycolysis was easy. And also when it comes towards the ends, you know that it's only itching to give us 2 more ATPs, right? So it's simple. Write down the 8 steps. Number 1. The first step is condensation. I think this is not given in detail in NCERT. I remember reading NCERT a few weeks ago where I was criticizing about I don't have NCERT. Can someone just look up at this Krebs cycle in NCERT and can count the number of steps and tell me? So first is condensation. Have they explained the steps or not? Second is dehydration. Condensation, dehydration, hydration. Take out water, put in water. Condense, take out water, put in water. Oxidative decarboxylation. Oxidative decarboxylation. Then followed by another, I think, oxidative decomposition, another oxidative decarboxylation. Same step happens twice for two different molecules. Then phosphorylation is the sixth. And you can now understand just by the name here that if I'm phosphorylating something, that I will get ATPs, right? Energy will come out. Then again, dehydrogenation. And then dehydrogenation, is it ulta? No, it's dehydrogenation, hydrogenation, dehydrogenation, hydrogenation. And then there is again, which is called, oh, I made a mistake actually. This part of dehydration and hydration is actually, sorry. I'm so sorry, people. Let's do one thing. So it's called 2A and it's called 2B. You dehydrate, then you put water back in, and it's 3, 4, 5, 6, 7. And the last, we end the cycle with dehydrogenation, where we make the product back again. So just write this and find your own ways to remember it. The way I remember it is we condense, sorry, what did I do? Condense, dehydrate, put water back in, oxidative decarboxylation into 2. Then phosphorylation, dehydrogenation, hydrogenation, dehydrogenation. This is the major step. These two are happening to make some changes in the molecule, and then these three are again happening to come back to the initial step. Okay, so all these steps I will teach you. Did someone looked up at Krebs cycle in NCERT? Are these steps explained?
They're not in very detail.
But are these steps mentioned?
Yes, sir.
Okay, well then very good. Okay, we'll go to the details. So we just have 10 minutes, 12 minutes. So I think let's take it in next class. Okay, tomorrow's class, right from the beginning, Krebs cycle. And we'll finish it. And we'll also finish fermentation tomorrow. So we'll try to finish respiration and then I'll tell you. Okay. Any doubts, people?
No doubt, sir.
Reba and Abhinav. Others, I think I've left. It's already night. Okay, then I declare the class and see you tomorrow. Good night.
Thank you, sir. Bye, sir.
Bye-bye.